Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision.
Right, like a broken bone or something.
Exactly. Like you break your arm, the X-ray shows a jagged white line, and the doctor just points right at it and says, there's the problem.
And here's the cast that fixes it.
Yeah. But if you step into the world of oncology, specifically advanced gastric or stomach cancer, suddenly that X-ray machine feels like it's totally broken.
Yeah, the landscape is incredibly murky.
It really is. I mean, picture two patients for a second. They had the exact same type of advanced stomach cancer.
Okay.
And they received the exact same cutting-edge treatment regimen.
Right.
But, their body's responding completely opposite ways.
Which happens a lot, sadly.
Right. One patient's tumor shrinks and stays at bay, while the other's just aggressively pushes forward.
It's really one of the most profound and frustrating medical mysteries we face today.
I can imagine.
Because we've historically treated cancer as a simple equation, right? Like drug plus tumor equals cure. We viewed the tumor as just this lump of rogue cells.
Just a static target.
Exactly. But the reality which we are finally mapping out in detail is that a tumor is a highly complex, fortified ecosystem. I mean, it has its own defensive walls, its own supply lines, its own chemical language.
And today, we're taking you, the listener, right into the center of that ecosystem.
We really are.
We're doing a deep dive into this fascinating new study. It was published in Cell Reports Medicine in 2026 by Wright and colleagues.
It's a fantastic paper.
Yeah, we're going to track the microscopic choreography of your immune system to see exactly how these cellular soldiers communicate to mount a successful anti-cancer response.
And there are some huge surprises in there.
Oh, massive surprises. Maybe most surprisingly, we'll discover how traditional old-school chemotherapy is actually setting the stage for modern immunotherapy to do its job.
Yeah, to understand why those two hypothetical patients diverge, we have to look at the clinical trial the study is based on.
So lay out the setup for us.
Right. So the researchers followed 33 patients with advanced gastric cancer. But they didn't just give them a drug and wait to see what happened.
Right.
They used a sequential therapy approach. First, the patients received standard chemotherapy, a combination of 5-FU and a platinum-based drug.
Okay, standard stuff.
Yeah. Then, for the following cycles, they received that same chemotherapy, but combined with an immunotherapy drug called pembrolizumab.
Which is an anti-PD-1 drug.
Exactly. It basically takes the molecular brakes off the immune system.
Okay, I want to pause on that trial design for a second because, well, from the outside looking in, it seems kind of counterintuitive.
How so?
Well, we hear so much about immunotherapy being this, ultimate modern sniper rifle, targeting the immune system to fight the cancer naturally.
Right.
So why are we starting the treatment with the blunt, explosive instrument of traditional chemotherapy?
It's a great question, and it really gets to the heart of how our understanding of these drugs is shifting right now. Because we used to view chemotherapy strictly as a cytotoxic agent.
Like a poison.
Exactly, a poison designed to kill rapidly dividing cells. But recent massive trials like Checkmate 649, they showed us that combining chemotherapy with immunotherapy improves survival significantly.
Even if the responses are still unpredictable from patient to patient.
Right. So the researchers in this new study wanted to know why. Why is that combination so potent? And to figure it out, they took serial biopsies.
Meaning multiple samples over time.
Yeah. They sampled the tumors before any treatment, then again after just the chemotherapy, and a third time after the chemoimmunotherapy combination.
And what they did with those biopsies is where the technology gets just incredible.
It's mind-blowing.
For you listening, they used single-cell RNA sequencing and T-cell receptor or TCR sequencing on over 66,000 individual immune cells.
66,000.
Yeah, across 3 time points. And think of RNA sequencing as dropping a microscopic microphone into the cell.
I love that analogy.
Because, DNA is the static blueprint, right? But RNA is the active transcript.
Yeah.
It tells us exactly what genes that specific cell is reading and acting on at that exact second.
And the TCR sequencing is like a GPS barcode.
Exactly.
It identifies a T cell's unique lineage. So that allows researchers to track that exact cell and its clones over time across all 3 biopsies.
So by tracking those 66,000 cells, they were able to divide the patients into 2 distinct groups based on how they did clinically.
Right, you have the slow progressors.
The durable responders.
Yeah, these are the patients who stayed progression-free for more than 6 months. And then you have the fast progressors.
The non-responders.
Sadly, yes. their cancer broke through the treatment in less than 6 months.
And the RNA data from those serial biopsies proved something really remarkable about that initial round of chemotherapy, didn't it?
It did. It showed that the chemo wasn't just killing tumor cells, it was actively remodeling the tumor microenvironment.
It was radically altering the landscape to prepare it for an immune invasion.
Terraforming the cancer, essentially.
Terraforming, yes. But who are the soldiers actually stepping onto that newly terraformed battlefield?
Well, the study narrows in on the assassins of the immune system, the CD8 positive T cells.
Okay, right. But inside a tumor, a T cell doesn't just have one mode of operation, does it?
Not at all. The researchers use a computational algorithm to group these T cells into distinct biological neighborhoods based on their RNA transcripts.
And they found two critically different states of T cells.
Exactly. And these states heavily dictated whether a patient became a slow progressor or a fast progressor.
So let's unpack the first one.
The first is the terminally exhausted T cell, or T-tex.
Okay, let's explain what exhaustion means here, because it's not just like a cell getting tired from a long day at work, right? It's a very specific biological state.
Yeah, in immunology, exhaustion is a programmed state. It's caused by sustained exposure to a threat, like a chronic virus, or in this case, cancer.
So the T cell realizes the threat isn't going away.
Right, and it scales down its response to prevent massive collateral damage to healthy tissue.
Oh, wow.
But these terminally exhausted T-tex cells, they are still highly cytotoxic. They are loaded with cell-killing proteins like perforin and granzyme B.
But their RNA transcripts show high levels of terminal exhaustion markers.
Exactly. They fight incredibly hard, but they have hit a developmental dead end.
Meaning they can no longer divide or replicate.
Right. They are done.
So it's like a war zone, and those terminally exhausted cells are your frontline infantry.
That's a great way to put it.
They're right in the trenches, firing their weapons, doing all the heavy lifting, but eventually, they run out of ammo and perish.
Yeah.
They're an essential part of the attack, but mathematically, they are a short-term solution if they can't be replaced.
Which is why the second T-cell state the researchers identified is the true linchpin of a successful response.
And what's that one called?
The progenitor-exhausted T-cell, or TPEX.
TPEX, got it.
These cells express a vastly different genetic fingerprint, like they express granzyme K instead of granzyme B. But the most crucial part of their signature is a master transcription factor called TCF7.
Okay, let me jump in here, because transcription factors are the proteins that physically bind to DNA to turn other genes on or off, right?
Thought on.
So what is TCF7 doing for these specific T cells?
Well, TCF7 basically holds the chromatin open for stem-like genes.
Oh, wow.
Yeah, it gives the TPEX cell the power of self-renewal. These cells don't rush to the front lines to die.
They hang back.
Exactly. They hang back and continuously divide. They generate a steady stream of new terminally exhausted cells to send into the fight.
Okay, building on that infantry analogy, these TCF7 expressing TPEX cells are the generals.
Right.
They're safely stationed at base camp. They maintain the supply lines of fresh troops.
That's exactly how it works. And when the researchers looked at the clinical outcomes, the data was just crystal clear.
What did they find?
The slow progressors, the patients who survived and fought off the cancer, had a much higher abundance of these TPEX generals.
And it wasn't just about having them initially, right? It was about their persistence.
Yes. Through all 3 biopsy stages, across months of harsh treatment, those specific self-renewing clones stuck around in the tumor.
And in the fast progressors.
Those base camps completely dwindled and vanished.
Man.
It completely shifts how we have to think about immunotherapy. For a long time, the goal was just to flood the tumor with as many active, highly lethal killer cells as possible.
Just throw infantry at it.
Right. But this study demonstrates that a durable clinical benefit relies almost entirely on preserving that reservoir of stem-like T cells.
Because without the generals constantly replenishing the ranks, the frontline infantry simply burns out.
And the cancer takes over.
Okay, so let's do a quick milestone recap for everyone listening. Checkpoint 1.
Sounds good.
So a successful, durable immune response requires a persistent base camp of these self-renewing TPEX cells inside the tumor. But this brings up a huge chronological question for me.
Let's hear it.
If these specific, highly lethal T-cell clones are establishing a base camp in the slow progressors, are they just multiplying locally inside the tumor from the very beginning? Or are reinforcements arriving from the outside after the treatment starts?
You know, the researchers asked that exact question, and to answer it, they didn't just look at the tumor biopsies.
They looked at the blood?
They looked at the blood. They took peripheral blood samples that perfectly matched the timeline of the tumor biopsies.
Oh, that's so smart.
Yeah, and they ran that same T-cell receptor sequencing on the blood to find the unique barcodes of the T-cells circulating in the bloodstream.
And then cross-reference them with the barcodes found inside the tumor over time.
Exactly.
This is the part that contradicts everything we often assume about how these cells operate. So where were the reinforcements coming from?
Well, in the slow progressors, the specific T-cell clones that flooded the tumor after immunotherapy was administered, they were actually already circulating in the patient's bloodstream before the treatment even started.
Wait, really?
Yes. They were present right after the first round of chemotherapy.
But hold on, if these highly targeted tumor-reactive T cells are just cruising around in the bloodstream for weeks, why are they ignoring the tumor?
That's the million-dollar question.
I mean, if they recognize the cancer, why aren't they leaving the bloodstream and going in to attack it?
Because a solid tumor is effectively a walled fortress.
Bull.
The blood vessels that feed a tumor are highly abnormal, they're chaotic, they're leaky, and they lack the proper adhesion molecules that T cells use to grip the vessel wall and exit into the tissue.
So they literally can't pull over and get off the highway.
Exactly. And on top of that, the extracellular matrix, the physical scaffolding of the tumor, is incredibly dense and pressurized.
So the circulating T cells recognize the enemy, but the physical environment is completely impenetrable.
Right, which perfectly explains why that blunt instrument of chemotherapy was necessary at the very beginning.
Oh, because the chemo acts as the breaching charge.
Precisely.
That makes so much sense.
Yeah, when those toxic chemo drugs cause a wave of initial cell death in the tumor, it triggers localized inflammation.
And what does the inflammation do?
It alters the blood vessels. It normalizes them just enough to express the chemical flags that let T cells dock.
And it softens that dense matrix.
Yep. The chemo creates the literal on-ramp, a permissive environment that finally allows these pre-existing circulating TPEX clones to infiltrate the fortress.
So they get in.
They get in, establish their base camp, and then the immunotherapy arrives to remove the molecular brakes, allowing them to mount a sustained attack.
It's an incredible 1-2 punch. The chemo blows a hole in the fortress wall to let the generals establish their base camp, and the immunotherapy arms them.
Exactly.
But, you know, once those T cells get inside, they face a completely new problem.
Oh yeah, it's not a friendly environment.
Not at all. A tumor microenvironment is brutal. It is hypoxic, meaning there's very little oxygen.
It's highly acidic.
Right. And it's starved of nutrients because the cancer cells are just aggressively consuming everything in sight.
Yeah, a T cell cannot survive in that environment on its own, let alone continuously divide and fight.
They need backup.
Desperate.
And this is where we meet the unsung heroes of this entire biological drama, the B cells.
This was perhaps the most surprising revelation in the entire study.
Really?
Yeah, because historically, oncology has focused almost exclusively on T cells when studying cellular immunotherapy.
Sure, because B cells are usually known for like sitting in the lymph nodes and pumping out antibodies against viruses, right?
Right. But this study revealed dramatic B-cell dynamics right in the middle of the tumor.
Okay, so what did they see?
They found that in the slow progressors, the abundance of B cells, specifically a highly active type called germinal center B cells, increased steadily throughout the entire course of treatment.
And in the fast progressors.
These B cells were virtually non-existent.
Wow. So why is the presence of B cells making the difference between life and death for these patients? What are they actually doing for the T cells deep inside the tumor?
To map this out, the researchers utilized a computational tool called Cell Phone DB.
Cell Phone DB, that's a great name.
It's very fitting because it doesn't just look at what genes a cell is expressing, it infers how different cells are communicating.
Okay, how does it do that?
It looks for complementary pairs. So a specific chemical signal being broadcast by one cell and the exact corresponding receptor for that signal expressed on another cell.
Oh, that's brilliant.
Yeah, and by running the RNA data through this tool, they uncovered an elegant, highly specific chemical conversation between the T cells and the B cells.
So what's the message?
Well, the TPEX cells are generals at base camp. They secrete a chemokine beacon called CXCL13.
Okay, and a chemokine is basically a chemical scent trail. So
Yeah.
the T cells are broadcasting this CXCL13 scent out into the surrounding tissue.
Right. And it turns out the B cells express a receptor called CXCR5.
Which is specifically designed to detect CXCL13.
Exactly. So the B cells follow that concentration gradient, the scent trail, right through the dense tumor tissue, navigating directly to the T-Pex cells.
And when they finally find each other, they don't just, wave and pass by.
No.
They engage in a physical interaction that is critical for survival.
They do. They engage in what's known as costimulation. The B cells physically lock onto the T cells using complementary surface molecules.
Like puzzle pieces.
Exactly. Specifically, a molecule called ICOSLG on the B cell locks into ICOS on the T cell.
Okay.
But more importantly, a molecule called CD80 on the B cell binds directly to CD28 on the T cell.
All right, I want to really visualize the mechanics of this.
Go for it.
Think about that fighter jet operating deep in enemy territory. The T cell is an advanced jet doing massive damage, but it's operating in a resource-starved environment.
Very starved.
So it's running out of fuel and nearing the point of crashing, or in biological terms, undergoing apoptosis.
Right, programmed cell death.
But before it fails, it turns on its radio and broadcasts a distress signal.
Yes.
That's the CXCL13 scent trail.
Yes.
And the B cell acts as a massive mid-air refueling tanker. It has its receiver, CXCR5, scanning for that specific frequency.
I love this.
So it flies directly into the hostile zone, locates the jet, and physically plugs into it. That physical docking, the CD80 locking into CD28, isn't just a signal.
No, it's not.
It triggers an actual intracellular survival cascade inside the T cell. It pumps life-saving biochemical fuel into the jet, preventing it from dying and giving it the energy to keep cloning new infantry.
That analogy perfectly captures the biology at play. I mean, spot on. That CD28 signaling cascade is what prevents the T cell from succumbing to the toxic environment.
It literally saves its life.
Yeah. The slow progressors survive because their immune systems successfully build and maintain this intricate, localized refueling infrastructure.
And the fast progressors fail because...
Because even if the chemo successfully lets T cells into the tumor, they lack the B cell tankers to sustain them.
Oh, man. So they just burn out and die.
Exactly. The communication data showed that the interaction strength of that CXCL13 to CXCR5 connection was massively higher in the slow progressors.
Heavy evidence right there.
Very heavy. It points to this exact mechanism as a primary driver of prolonged survival.
Okay, let's do our second milestone recap here.
Good idea.
This fundamentally changes how we view an immune response.
Right.
Like, it's not just a wave of single cells operating independently. It is a highly coordinated logistical supply chain.
That's exactly what it is.
You need the T-Pex generals to maintain the ranks. You need the chemotherapy to terraform the blood vessels so reinforcements can arrive. And you absolutely need the B-cell tankers to navigate the chemical gradient, physically dock with the T cells, and keep them alive in a toxic wasteland.
When we pull back and look at the broader implications for the patient, it's a massive paradigm shift.
I mean, it proves that treating cancer cannot be about targeting the tumor with a single magic bullet.
No, we have to practice ecosystem management. We have to reevaluate traditional chemotherapy, not as a blunt poison, but as an ecosystem remodeler that prepares the soil.
Which completely flips the script on how we view chemo.
It does. And it shows us that the future of immunotherapy isn't just about unleashing T cells. It's about ensuring the entire immune neighborhood is present and communicating effectively.
You know, it's just a testament to the staggering complexity of the human body.
It really is.
I mean, think about what is happening inside you right now just to maintain your health. Millions of different immune cells are perfectly coordinating their actions in space and time.
It's beautiful, honestly.
It is. A T cell has to sense a toxic environment, manufacture and secrete a specific chemical scent trail, and a B cell has to detect it, travel through the bloodstream, squeeze through a tissue barrier, navigate a hostile tumor, and physically lock proteins together just to keep the defense from collapsing.
And thanks to technologies like serial single-cell sequencing, we are no longer guessing why this complex system fails or succeeds.
We can actually see it.
We are literally reading the communication logs of the immune system as it fights. We are mapping the exact vulnerabilities in the tumor's ecosystem.
Which leaves us with a totally fascinating question for the future of medicine.
Oh, absolutely.
Because we've spent this entire deep dive tracing how critical this B-cell refueling infrastructure is to keeping a patient alive.
Right.
So could future therapies exploit this directly?
I think that's where the field has to go.
I mean, think about the implications. If B cells are the ultimate survival mechanism for T cells, what if we stop focusing solely on engineering better, more lethal T cells?
Right. What if we focus on the supply line?
Exactly. What if instead we inject synthetic CXCL13 directly into a patient's tumor?
To call in the B cells.
Yeah. Or what if we could engineer super B cells in a lab that are hyper-responsive to a tumor environment?
That would be game-changing.
Right. Could we intentionally turn any cold, unresponsive tumor into a highly attractive, welcoming refueling station?
We could essentially trick the body's natural immune system into swarming the cancer.
Simply by building better supply lines before the battle even starts.
It changes the entire narrative of oncology.
It really does. It's definitely something to mull over the next time you think about how your body heals and adapts.
Oh, new perspective.
Well, thank you for joining us on this deep dive into your own cellular ecosystem. And as always, keep questioning the world around you.